Okay, so those thermal treatments we talked about, denaturation condition at 94 degrees Celsius, that's around like a water boiling temperature, very hot. And then whatever that primer binding temperature is, that temperature should be calculated based on your primer sequence. And then polymerization temperature 72 degrees, that's also a hot temperature. If you stick your finger in there, you're going to get a skin burn there. So these three different temperatures should be alternated very rapidly, so your reaction is not going to take several days, right? So you need a very efficient heater and maybe the cooler that can change the temperature. Maybe they can ramp up, ramp down the temperature very quickly. So that function is done by this little machine called thermal cycler. Okay, so inside this box, if you open it up, there are these tube holes where you can place your tubes. So that heating block will have a controlled temperature. So it's heating up, heating down very efficiently. So this is a very efficient heater and cooler. You can program the temperature and time. So that's a thermal cycler. Okay, that's how your PCR will be done. Okay. So let's talk more about the denaturation happening at the high temperature. So during the time denaturation condition, complete strand, the DNA template strand, will be separated into singles, right? It's a very critical step. Without the denaturation, your primer would not be able to bind on the target site, right? And then generic method recommends about 30 seconds of treatment at 94 degrees or 95 degrees. That should be fine, okay? And then depends on how many GCs are found from your template DNA. You may have to extend that temperature condition a little bit, but normally, 30 minutes or 1 minute is enough. Okay. And the second step, like I said, is a primer binding or annealing step. So primer will be finding the target and binding. Okay. So what temperature condition are you going to use for that? So primer annealing should be done at the correct temperature. So it's not too high or too low. If you're doing it at too low condition, something called random binding may take place. So primers, not only they can bind on the site with the perfect sequence match, they can also bind on other sites with maybe 50% sequence match or 30% match, 40% match. Okay, we call those random binding sites. If primers are binding on those sites, your PCR the replication will give you random products that are not within two primer binding sites, and that's going to be a problem, right? So to eliminate those random binding, you want to crank up the temperature. So when you heat up the temperature at high condition, those random binding site, they will lose the primer because their hydrogen bonds will be broken up. Okay. So then what is the best temperature? You need to calculate it out. So first you need to find out at what temperature your primer would fall off from your target site. So that is called a melting temperature. Okay, so you need to mathematically figure out what the melting temperature for your primer is. Okay, so by looking at the primer sequence. So what is the secret for that? We're going to talk about that. Let's see. Yeah, here is the equation. Okay, so to determine the TM melting temperature, this is the equation you use. So count all the G's and C's that are found from your primer sequence. Let's say within your primer there were 12 G's and C's, and 12 times 4. Okay, so that's about what? 48. Okay, and then count the number of A's, T's that are found within your primer sequence. Let's say there are 8 of them. Okay, 8 times 2 is 16, right? 16 plus 48 is what? 50 or 64, whatever, right? 64. Okay, so the 64 degrees will be the melting temperature of your primer. Okay, that's how you can figure it out. So once you understand the melting temperature, okay, then normally melting temperature minus 5 degrees your binding temperature. Okay, so you need to do the primer binding at that temperature. Okay, so that's the key. And then once you are done with the primer binding, and then you're going to do the extension. Extension is the condition where the DNA polymerase, in this case Taq DNA polymerase, will bind at the tail of your primer, and then it's going to start the polymerization by adding all nucleotide on the newly made strand, right? Then why do you do that at 72 degrees Celsius? Like I said, the polymerizer you're going to use a Taq, which is isolated from the extreme thermophile. Okay, so the 72 degrees in Celsius is the optimum condition. So normally 1 minute at that temperature will be good. So ramp time is how much time does it take for this thermal cycle to move the temperature from like a denaturing condition to the primer binding condition, from the primer binding condition to the extension condition. Okay, so those are the ramp times. Okay, so if it's an efficient thermal cycle, the ramp time will be very quick, very short, so you can complete 30 PCR cycle within at least a couple of hours. Okay. So the primers are important in your PCR reaction, of course, because primers will designate which part of the template DNA you will amplify out. It's between two primer binding sites. That's where the PCR replication will take place, right? So primers will recognize the target sequence and they'll bind to them, okay? And then two primers will designate the region of amplification, okay? So they're very important. And then they may have some random non-specific background binding. And you can minimize those non-specific random background binding site by doing the primer binding at higher temperature. Okay. So the region of amplification, it could be flexible. Maybe it's up to 10 kilobase pair. Okay, but normally it's done around like a 3 kilobase pair size. Okay, and the primer themselves, the size should be between like 20 to 30. Okay, so about 21, 25, those are fine. You're going to do a lab 6 on primer designing, so you'll get to learn more about how you select a good primer binding site and how you analyze primer sequence for their secondary structure and things like that. Okay. And the stability of a hybrid between the template and primer is expressed at the melting temperature. I told you, that's the temperature condition where the thermal energy is great enough to break all those hydrogen bonds between the primer and the template DNA. So you don't want to do your primer binding at the temperature. Instead, you want to use the temperature 500 degrees below the melting temperature. Then why do you need to know the melting temperature? Because you want to use a higher primer binding temperature to eliminate all those random binding sites. Okay. So several computer programs can help your primer designing. Again, OligoAnalyzer is the computer software you'll be using in the lab 6. You'll get to do that. Okay. And then primers may have intermolecular complementary. What does that mean? It's a difficult to understand word. Intermolecular, what does that mean? Between two molecules, complementarity. If the molecules have some complementary sequences, they will interact. That's what this means. Okay. For example, so there's a primer. Okay. If that primer has matching sequence with another primer, okay, this primer and another primer, they can bind together. So this is an intermolecular binding between these two primers. Okay. So what does that mean? So if the primers are binding between themselves, primer 1 and primer 1, okay, that primer 1 is not going to bind on its target template DNA. Instead, they'll be fully occupied among themselves. Then that means that the bad news, because primers are designed to bind on the template DNA, not by themselves. So you don't want to use a primer like this. So the primer should not have a lot of these intermolecular complementarity. So they will bind by themselves, right? You don't want those. You want to avoid those, right? And the primers can also have intramolecular complementarity. What does that mean? Within a single primer, it can form this unique hairpin structure because the primer itself has some complementary sequence matches within it. Okay, so that's the structure called hairpin or intramolecular complementarity. So hairpin is based on its intramolecular complementarity sequence, right? So if your primer exists as this form, would it good to be able to bind on the template DNA? No, it's not good either. So you don't want to have a primer with a bunch of these sequences. So a primer without the hairpin structure formation is a good primer actually. So you want to avoid that one as well. So your primer analyzer program actually look at the primer sequence and identifies all these hairpin structures on as well as all these primer dimer formations and let you know that you should not use this primer if there are too many of these formations. Okay. And then mismatched primers used for some amplification. What does that mean? Intentionally you can make a primer with a mismatch. Okay, this primer with a single mismatch, that primer with a single mismatch is binding on its target DNA, and let's say that target DNA has a gene sequence there. And when this primer is amplifying around, you see the products there. Okay, and those two products can be separated out, and they can anneal by themselves. Okay, and then those gaps will be sealed up, and you can make a new double-strand DNA with one nucleotide mismatch from the original template DNA. So what's the importance of that, and why do you want to do that? One nucleotide mismatch, because that is used for your site-directed mutagenesis. Okay, one codon with one nucleotide change. Okay, that codon will call out for different amino acid within the protein that is encoded by this gene. That's used for site-directed mutagenesis method, using these primers with a particular specific mismatch sequence. Okay. And then template DNA is important, especially the quality of it, when you use that DNA for your PCR. Because, if the DNA is dirty, it has a lot of contaminants. Those contaminants may hinder your primers binding on the template, and as well as the RNA polymerase doing its job. Okay, so your template DNA should be clean. So you should not contain a lot of denaturants like urea, STS, sodium acetate, and gel debris from your purification process. Okay, so you make sure that you're going to clean up your DNA by doing ammonium acetate precipitation with the ethanol presence. Okay, so you use a clean template DNA. That's important. And then Taq polymerase, like I said, is a unique polymerase that can withstand the heat. So why do you have to use Taq polymerase instead of normal DNA polymerase? The reason is, how many times are you repeating the PCR cycle? You repeated about 30 cycles, right? So your DNA and your Taq enzyme will be exposed to the fluctuation of the temperature about 30 times. Heating and cooling, heating and cooling, heating and cooling 30 times. So if you use a normal DNA polymerase it cannot go through 30 times of heating at 94 degrees, right? But this Taq polymerase can withstand that treatment. So that's why you want to use this Taq polymerase, which is heat-resistant DNA polymerase, for your PCR thermal cycle. So Taq polymerase can withstand the 30 treatment of the denaturation process. Okay, that's why you want to use a Taq. Okay, but Taq polymerase, they tend to have a higher rate, a higher error rate, meaning every about 20,000 nucleotide amplifies, there may be a error, one mistake it can make, and it also doesn't have the auto reading function. So it's a kind of disadvantage of using the Taq DNA polymerase. Okay, and Taq DNA polymerase, by its design, also tend to add one extra nucleotide at the end of its PCR cycle. So that extra nucleotide is actually extra A. It's adding on its 3' end. So actually your PCR product will have one extra A overhanging on the 3' end of each DNA strand. So A overhang is normally made in your PCR product. So is that a good thing or bad thing? It may be a good thing if you're going to use that feature in your cloning. Your PCR product can be easily inserted into a plasmid DNA that has, T overhang, if you can make the one. Actually, that PCR cloning plasmid DNA is available. It's called TA cloning PCR plasmid. So by using that T and A match, you can easily insert your PCR product into the plasmid DNA. So that will be a nice cloning. So that can be used. So that's a unique feature of the Taq polymerase. It adds extra A at the end of the 3' end of the PCR product. Okay.